grateful to Professor Alberto Diaspro for agreeing to deliver the inaugural talk. And his CV is vast, so I can't really, I don't want to go through it. It's all over the internet. You can find it. And he has immensely contributed to the ever-changing field of optical microscopy and imaging. Today, what we call as a nanoscopy. And his spectrum is really large, from light sheet to confocal, two-photon, single molecule, super resolution. I can just keep going on. At present, he is one of the giants of the field and well respected around the world. In short, he has several international awards, recognitions, presently serving on many editorial boards of respectable big journals. And I don't think there is anyone better who can explain the microscopy and the contribution of microscopy for humankind. So I believe, once this talk is over, we will see the contribution that the field of microscopy has, on the humankind. So what I'll do, I will ask Professor Alberto to really address us and talk about this fascinating talk, that he's going to deliver now. Professor Alberto, please, you may please go ahead.
Thank you very much, Partha. Thank you very much, everybody, for joining this lecture. And I have to say, just let me comment just for a while that it was great having you in the lab as postdoc and our collaboration. And I'm very proud to be today in the meeting and in this series of lectures you are organizing. So now I'm going to share my screen so you can so I can start my presentation. Let me see if I can do this. Okay. I assume you can see my presentation, right? Can you hear me?
Yes, Professor, it's visible.
Okay. So if you see my screen, I can start. Okay. Partha, can you see the very first slide? I don't.
Yes, we are able to see and hear you.
Okay, so thank you very much. So the title I suggested, I proposed to Professor Mondal, is the intelligent optical microscope at the nanoscale from cells to molecules. So let me try to go into this story. But before, this is something that in Italy we do, and all over the world we ask that you do. We are asking for truth for this researcher. His name is Giulio Regeni, and he was killed by police in Egypt, and are 7 years that we are asking for justice. So please join us in this request if you can. But now let me go to microscopy, and the very first slide is related to my group, the group that was also populated by with part of Pratim Mondal many years ago, and he provided a very good contribution to our research and developments, and this is the new group. And so if you see things you like, they are mainly responsible for what you will see in the next. Let me also start with this slide again to remember Tullio Pozzan, that was an Italian scientist, very active in microscopy, very active in neuroscience, one of the few Italian scientists in the National Academy of Science of the United States. And so this lecture is dedicated to the memory of Tullio Pozzan. And now, this is my very first slide that is bringing to you the complexity of an architecture that is trying to collect everything we are able to do in microscopy in order to get the maximum in terms of information from the biological samples we are interested in. I will be back on this slot by slot, of course. As you know, as you do probably, we deal with a very small portion of the electromagnetic spectrum that is the visible spectrum, visible light. There are many reasons for dealing with visible spectrum, and one of them is related to the fact that biological macromolecules do not absorb light in the visible spectrum. In case they absorb, except with some exception, I mean, that are very well protected in the human body, like hemoglobin and some others, but in most of all of our biological macromolecules, they do not absorb. We are very lucky about this, because in case they absorb, we would have a very high increase in terms of temperature. So this is a lucky case. And we decided to use the optical microscope in order to perturb at the minimum level biological macromolecules. So we shine light and we try to collect information. Shine light. What does it mean? It means that I need to have in mind something related to illumination sources, and then I have to find a way to get information from the interaction, and then to try to build an image. This is what we like to do. I don't know if you know the myth of the cavern by Plato, that was a philosopher, but the idea is that you are in a cavern and you are in front of a screen, and behind you there is light, that is the knowledge, and things moving, and you try to understand what's going on. This is what we do mainly with the optical microscope. And if you think about pictures that you take with your camera, there is something that is different when you consider a photography or an image formed at the microscope. Going to the photo is related to the real world. There is nothing that you can add when you click with your camera, but what is relevant is this sentence by Gianni Berengo Gardin, a very famous Italian photographer. You walk into a place, you see there could be a great picture, but something is missing. If that something comes, you can get a good photo. If not, it's nothing. What does it mean? This means that when you have your problem, when you decide to take your picture to form your image, that is not so immediate like when you take a picture with a camera, you need to be able to see something that at the very beginning you were not able to see. This is our job. This is a very famous picture by Gianni Berengo Gardin, and so this view, this image brings you in something that you could not imagine. A boat in the city of Venice is not, but this is what appears in this picture. And if you get this sport picture, there was a debate since 1952, who was the one passing the bottle to the other. You cannot solve this problem by this picture. You have to add information to solve this problem. And one of the best ways for adding information is collecting pictures, collecting information also in time. Like in this painting of the Cathedral of Reims. This is an impressionist artist. Or you can also use your camera to take pictures in motion, like in this way. This is done by purpose. And this guy, Roberto Polillo, does something that is as some inspiration for people working in microscopy. You keep your camera, you have your camera, you have what you want to get with your camera, the picture you want to get, and then you decide to move your camera in time while something happens. You add information and you add your own view of the situation you are trying to freeze. There is a movie, I don't know if you have this movie in Bangalore, called The Fabelmans. And in this movie, what is nice is that this is the story of a mother providing an instrument for capturing image to a boy, and this boy is fascinated by illumination and by the chance of bringing an image somewhere in his hands. This guy is Steven Spielberg. And when you shine light and you bring light to your object, there is a new world that opens its door. When I told the story of what I wanted to do with the instrument, you have seen in the very first slide that is a multimodal microscope, and MOMICS means multimodal optical microscopy image correlation sensing. This graphic artist, Marta Fioravanti, decided to give me this painting, a brain thinking the problem, the image, the potential solution, some light on the left side, colored light, visible light, some biological issues, and in the middle, a new way, a new part of our microscope, illumination, the objective, the sample, and some artificial intelligence, some intelligence in the middle. This is the reason why we are going through this. Now, if you consider the microscope as an instrument for capturing images, you have also to consider that you can be in the situation to perform what is called perfect imaging, because our objects are transparent. This means that the refractive index has only its real part, so you have no absorption, and so you are in trouble. So how can you make the contrast? I mean, you have this perfect glass, completely transparent. In the past, in 1906, an Italian, Camillo Golgi, got the Nobel Prize, was the first Italian to get the Nobel Prize. In the very same year, there was also a poet, Giosuè Carducci, Italian, getting the Nobel Prize, so two Italian in the very same year, for finding a way for bringing to you the invisible information of neuron in the brain. Amazing. And later, Fritz Zernike introduced phase contrast to transform the phase delay, the phase-induced delay, in an intensity image. Great. But now we are also able, and we learned how to decorate our sample to see information by colors when we shine light. And this is fluorescence, you know very well. And I'm very happy that with part of our team Mondal, we compiled this book, Fundamentals of Fluorescence Microscopy, to introduce people to use fluorescence. There is one of the pioneers in fluorescence microscopy, his name is David Jameson, that when you ask him why fluorescence, his first answer is because it's pretty. Come on, what does it mean? It means that there is some beauty for sure, but it also means that you can collect a lot of information when you shine light on your fluorescent molecule. Information related as function of the fluorescent probe to the number of ions, to the electric field around, viscosity, temperature, pH, polarity of the system. So really, using fluorescence, you can collect a nice image, not only a nice image, but an image full of information that you can use. And I have to say that the new science in fluorescence after Galilei in science is the one delivered by the pioneer and the master for everything in fluorescence, that is Gregorio Weber. And I strongly suggest you read this book, Perspectives on Fluorescence, a tribute to Gregorio Weber. Now, so, you know, with fluorescence, you can see a lot of things like these ones. You can see neurons in colors, and colors are of interest for the connection in the brain. You can photoactivate fluorescent molecule linked to specific part in the sample, like in this case, and what you are able to do, something that you were not thinking before. So you photoactivate molecules in some DNA region in a mother cell, and then when this mother cell divides, you can follow the fate of this, or you can decide to be very sharp in your system. This is two-photon microscopy, so it's a very highly localized form of microscopy, and you can photoactivate in a small region, the pink one you see in the image on the left, and then when molecules are out from this pink region and you detect them in the membrane, you can be sure for the localization of the event that they come from that vesicle. So we have really so many experiments we can do, and so many experiments we can merge. If you're interested in single molecule information, the only way for getting single molecule information, the real single molecule, direct on the single molecule, is performing some electrophysiology using patch clamp, or only in this case, you get information from single molecules. When you perform fluorescence, you get information from about the position of the fluorescent molecule. And you can use these to be more sharp in terms of deciding from which region of your sample, in this case a neuron, the information comes. So really using fluorescence, using a mechanism of contrast, using an optical microscope, you can make visible something that you cannot see, like in this case. Behind this, there is this word. And this word is a word that is a different scale, from centimeter down to nanometer. Today, you can really have a spatial resolution, ordinary spatial resolution below 100 nanometer, down to 50 nanometer, and down to tens of nanometer. But now, when I say something related to the space, I have to consider resolution and super resolution. And when you see this formula, I'm quite sure that you remember very well that this is the Abbe formula. Well, let me say that unfortunately, or fortunately, Abbe never wrote this formula. You can find the full story in this paper. He made an experiment using coherent light, not incoherent light, and he never wrote this formula. However, this formula matches very well with the results found by Ernst Abbe. So the optimal distance you can detect is related to the wavelength and is related to some properties of the matter, n, refractive index between your objective and your sample, and the geometrical characteristics of your objective, that is the alpha, that is the semi-aperture of your objective. So in this cone, half an angle. When we talk about resolution, we can talk about resolution in space or in frequency domain. This means that you can describe an object by superposition of sinusoidal functions at different frequencies that tell you which are the detail in the image. And when you go into a Fourier space, low frequencies are variation of intensity that are very smooth, and details are at the high frequency. Your objective, our objective, cuts the high frequencies. So you have limitation in detail, but how can you, we need a criterion for defining resolution. And in this case, you can model, you can perform an analytical formulation, you can use calibration sample like DNA origami that probably you know, or object where you put fluorescent labels at distances and nanometer scale that you know, or what we usually do is to trace a line profile and then to try to find a way for saying which are the points we are able to distinguish at a certain distance. Fine. The first method, you can make this sophisticated as much you want, but it's not the real world. Calibration sample is not the sample you want to study. And line profile is a subjective way of finding in your image the region where you want to detect resolution. What we propose in the last year is to use Fourier ring correlation to be more objective. This is very objective. I mean, what you need are two independent images. You take one and then you take another one in a very short time. In case objects move, don't worry, because in this case you will see this in the transformation, and so maybe you have an idea of resolution that is worse than the real case, but is better than overestimated resolution. You perform Fourier transform, you cross-correlate, and then at the cut with the threshold of noise, you have the effective cutoff of frequency. You go back in space, and you have the spatial resolution. This spatial resolution is independent from all aspects you can consider in this, from the sample, from the different alignment of the system. You don't need this kind of information. You simply state which is your resolution. And this is important now because, I don't know, okay. Because when you have a filament is comparatively easy to find a line, but when you have a situation like this one with a lot of molecules, it's difficult to say what's going on. And so if you perform Fourier ring correlation, you can provide a clear idea, in this case, 37 nanometer, for analyzing the image. This is what you need. You want to understand from this image what's going on. And you need it to be quantitative on the resolution you have at your disposal. Then, if you go through 1955, you have the very first declaration of super resolution made by this guy, Giuliano Toraldo di Francia, resolving power and information. What does it mean? It means that Toraldo di Francia has a seminal paper, I suggest you read it, telling you we don't want to fight against physics, so we don't want to overcome the diffraction limit. We have the diffraction limit, Abbe formula or whatever formulation you want. But if you are able to add other information before you form your image, you can get an image having a detail that exceeds the diffraction limit. This can be done, for example, in a microscope that probably you know that is a structural illumination microscope. I don't want to go into details in this, but very simply, what you are doing in the spirit of Giuliano Toraldo di Francia, you have a sample that is unknown. You create an illumination with known distances, known resolution, known frequencies, and then you provide the superposition of this illumination pattern with your sample. You have this moire pattern. You have a final image that combines the two, and you can extract the super-resolved information, like in this case, from the left to the right. This was the very bright idea by Mats Gustafsson, that unfortunately passed away very young. Then there was another paper, 11 years after the Toraldo di Francia paper, about the possibility of exceeding the classical limit of resolving power by Lucas. There is a very important statement by Lucas here. Please consider it and read the paper. What would you sacrifice in order to get a better spatial resolution? The answer is comparatively simple. You can sacrifice the field of view. So you shrink the information before it reaches the sensor. If you play this game by yourself now, you see better in front of you, and then you can scan in case you want to get the complete information. I know that you know that there are so many methods like the ones you can see reported here for taking advantage of a light matter interaction. We will discuss only some of them. And with Paolo Bianchini and Giuseppe Vici-Domini, we decided to offer the chance to use another method that is bringing more information together. And this is today an instrument called NSpark by Nikon or Prism by Genoa instrument. Simply, we decided to use a very comparatively old idea, and I will show you why, which one, to use multiple eyes to get the very same information from different views. This idea is called today image scanning microscopy and was a very bright idea by Mario Bertero and Roy Pike, 1981. Well, this was a theoretical paper at the end, having a very simple statement. When you shine light to a point, instead of collecting only the light using one sensor, you can use an array of sensors. And then Colin Shepard developed the theoretical approach to demonstrate this, and Jörg Enderlein, in this great paper on physical review letter, demonstrated that using an enhanced CCD camera, so a very nice collection of sensitive elements, you can get a super-resolved image. So instead of shining image and collecting from one point and forming your image, you shine light, you collect information in a region in your camera, and then you make some calculation to reassign the energy to the central point you illuminated. The main drawback of a camera is that you are limited by the frame rate. This is the reason why, with Giuseppe Vici-Domini and Paolo Bianchini, we started a kind of adventure in proposing to have an array of detector that is independent from the frame rate. And this is the SPAD array, single photon avalanche diode device. Multiple eyes. So when you shine, you collect not only the central point, as you see here, but also information from other points. You have to calibrate your system in order to reassign a light information and energy information that is provided by the point you illuminated in different regions with different views. If you're able to do this, you are adding information to your information channel, and so you can improve simply in this way resolution. Now, if you put your chair far from the screen, or you close a little bit your eyes, maybe you can realize what is behind this collection of pixels. Now, we collect an information like this, then we reassign information, and we get the real image with an improved resolution, like this, in this case, with this example. Now, if you have a look now to the very same image of before, now we have an additional information that is this one that you have now in your brain. So I'm quite sure that now you're better able to recognize what is behind this pixel. So adding information is useful for deciphering what's going on in our light matter interaction, and you can apply these to any kind of image formation, including the case of two-photon excitation. But what is very interesting behind this detector is not only that it's able to provide collection of information independent by the frame rate. You have single photon counters here, but also you can detect, in case you have a clock from the illumination with the time you collect the first photon, you are able to collect the time of arrival of photon. Time of arrival of photon is relevant because in fluorescence, for the very same color emitted, time of arrival tells you something about the neighbors of the fluorescent molecules. It's called the lifetime. And so adding spatial super resolution available by ISM to spectroscopic abilities, what you can do is to perform fluorescence lifetime imaging in a super resolved way. This is what have we done with Giuseppe, Paolo, and some other, and Colin Shepard in our lab. And in case you're interested in the characteristics of this sensor, these are amazing. I think that today, this is the best sensor you can have in terms of a sensor for image scanning microscopy. And we are also improving the ability of collecting light by realizing an array of micro lenses in front of the detector itself, so you can improve. And this is the reason why we decided to develop a startup called Genoa Instrument that, using image scanning microscopy in a very simple way, can offer you an improved resolution with respect to a conventional confocal microscope, adding the temporal super resolution. I think that this is a very nice example of bringing some new technology to a portable instrument like this one to perform super resolved image in a very fast way. You cannot reach the nanometer level, but you can improve your resolution, let's say from the upper limit, 250, down to 180 nanometer, simply using a very simple approach, also in terms of sample order. I think that this could be interesting. And then you can also use this for other potential application in healthcare, having a potential impact on the society. Recently, just this week, February 1st, so I bring this to you as a novelty, this approach has been introduced due to the relationship and work done with Nikon in the new confocal-based super resolved microscope by Nikon, and this is called N-Spark. The N-Spark detection is the detection we suggested with the SPAD array, and so they decided to implement this in the new generation of confocal microscopes that I think is the newest. So this is the traditional detector image. This is what you can get with the N-Spark, and this is the real comparison. There are other ways for improving resolution. For example, my granddaughter question, Irene's granddaughter's question, can we install polymer chains of a swellable material? What does it mean? Come on, I'm not crazy. It's not the Alzheimer now. There is a point driven by the Boyden group. Is not a hyper, is the fact that you can improve resolution if you're able to put at distances larger than the diffraction limit the objects you are interested in. I'm sorry, I see on my screen different notifications. I hope that you can continue seeing what I'm presenting. So expansion microscopy is a comparatively simple method. You put fluorescent molecule in your sample, you cross-link them with a gel, you break everything except the cross-links, or you can do this in a more gentle way with more recent methods that you see here cited. Then you put this in water, it grows, molecules go at distances larger than the diffraction limit, and so you can see them and you can bring them in your image, like in the case you see here with the microtubules. And we also demonstrated in a recent paper that we are able to perform this at the nanoscale, using the nuclear pores as a target sample. There are several advantages. You can see really things in an enlarged way, in a way that you couldn't see before using an optical microscope. Here is the comparison with the focused ion beam on the left upside with an optical image obtained after expansion microscopy. And so in case you are interested in this preservation of the expansion at the nanoscale, these are reported on this paper, appeared on Journal of Biophotonics, and these are some examples of the utilization of this. But now, in our scheme, there is also, of course, super resolution as the one provided by Betzig, Allen, Mueller, that got the Nobel Prize for this. I want to bring to your attention not only the images you see here that are super resolved images in terms of more details than I expected, but the motivation by the Nobel Committee. Motivation by the Nobel Committee is, sorry, I see other notification. I don't know what if I have to click or not, or you can do that for me. For realistic quantitative description at the nanoscale, resolution of the dynamics of the complex, multidimensional molecular biology processes that define the phenotypes of all life forms. So the ability of being super resolved open for us a new window in studying biological systems. There is something that I cannot control, that are these notifications that I see. However,
Alberto, we are hearing you loud and clear, no problem.
Sorry?
We are able to hear you loud and clear, no problem.
Okay, because I see this notification about Pratyush Bhushan in the list or whatever. However, the way for bringing additional information in the super resolved Nobel Prize are of two kinds. One is to be sure that you are dealing with single molecule, and in this case you can be more precise in localization with the square root of n, and n is the number of photons you are able to collect from each single molecule. You are able to select. This means that if you have 10,000 photons, you can improve to order of magnitude. The other one is that you can shrink locally in a very precise way the information coming from the sample that you excited, and this is STAD. We were able in the past to localize a single molecule and to produce images, but only when the single molecules were distances larger, like in this case, of the diffraction limit. The novelty in the Betzig approach, driven by the photoactivatable proteins developed by George Patterson in the Jennifer Lippincott-Schwartz lab, is by the fact that what you see here are molecules, the bright ones, proteins, that are photoactivated, so they can emit light, but since you photoactivate it with a very low dose of light, you have them at distances larger than the diffraction limit, so you can collect from each of them photons. And if you collect a large number of photons, you can move to the right side, bringing their position, more precise than using the diffraction limit, in your final image. So the final image you see on the right side appears like a cytoskeleton, common cytoskeleton image, except the fact that you know with the precision of 15 nanometer the position of each component of the alpha tubulin architecture in the cell. Of course, if you want to have a social impact, we have to bring this to real cases like a tumor, like a metastasis, like a tumor spheroid. So we needed to go on thick sample. And here is comparatively simple. I mean, what we demonstrated in this Nature Methods paper 2011 is that you can shine light in a different way, a plane of illumination. So you remove background from adjacent planes, and within each plane of illumination, you are able to detect single molecules from a large sample. And you can navigate through your object, detecting individual molecules plane by plane, increasing the thickness of the specimen that you can analyze up to 100 micron here, more than 200 today. Then if you go to STED, that is the other way, so you shrink the information that you were able to excite using your excitation, the final effect you get is that you have a brush, in case you want to consider yourself a pointillist painter, that is finer than the one you have when you excite. So what you simply do is this comes from the Stefanel lab, you provide your excitation, you have the molecule, excited molecule replying, then you superimpose another beam that shrinks the information for stimulated emission, and so you can get a better resolution in your image. So if you have this image and you shine the second beam, you get this one. And so you can perform this in real time without counting all the frames or detecting different single molecules. And you can see nuclear pores that you could see before in this way, as you have seen from other sample images. Or you can detect the entrance of SARS-CoV-2, the green, through a membrane of the cells infected by the virus. There are several examples. I don't want to go through all of them, just showing you some of them. But now I have to bring to your attention this image that comes from the Stellaris 8 tau STED. If you add another information that is lifetime to your intensity information, instead of getting, as in the gray left side, nothing from your sample because there is a lot of noise, you add another information that is lifetime point by point when you shrink the information with your STED, and you can detect objects that you couldn't see, adding another information. I've no time for going in details on this. I understand that I have, but you will have this slide. I will send this to Professor Mondal. I want to bring your attention to the final resolution. Today, there is a mix of single molecule and STED that was demonstrated in this Nature Photonics paper. And what is amazing here is that room temperature, normal pressure, you can locate, you can get resolution around 2 nanometer, and you can get a precision of detection around the angstrom level. This is the final, so no more limits, I would like to say. Just a flash on this, since now we have a very high resolution, what is relevant for us is to add also other methods for investigating the sample. And one of the most used methods in microscopy when studying biological systems is atomic force microscopy. And this is the reason why today, since you have a super resolution, please think to put them together in order to, for example, in this case, to understand from which real filaments that you couldn't see using the confocal, you are evaluating, for example, the Young modulus. And so you can also use this for understanding better what you're doing when you're labeling your sample. Sometimes you don't. Before, when you had 200 nanometer resolution, you could see something from your sample. But today that you have nanometer resolution, sometimes you don't see the fluorescent molecule. And you could argue that you don't have single molecules there. Something happened. Is it true? Or simply you were not able to put your molecule, that is a fluorescent molecule, in the place you expected? We decided to couple, again, atomic force microscopy to STED for trying to understand better what's going on when we label our sample. So the system we have at our disposal implements all the methods we are able to manage. And so we can face different biological problems from tissues to single molecules, like in this case. So we can study the meaning of switching on of cancer progression. We can study, in case of progeria, the effect of a genetic disease in the membrane, bringing to these patients that were born old and die very soon. Or we can study, for example, these are only 3 examples, but you can have in your mind and in your lab many others, neuroblastoma progression using a combination of the super-resolved approaches. Amazing, would say my granddaughter. Yes, amazing. And you can find this story in this paper that is open access on this international journal. But now there is a problem. And this is the other part, another part of my talk, that is the environment we're interested in is crowded. Fluorescent molecules increase crowding. I'm not sure this is relevant or not. But what not thinking about using super-resolved methods or optical approaches that don't need any labeling? Well, when you use two-photon excitation, you can excite autofluorescence within your sample in 3D, and moving the Z position, like in this case, you can detect new objects using intrinsic fluorescence or without labeling. We can play this game. And it's nice, the ability of performing this game in your sample. You see here, you see sometimes new objects appearing using autofluorescence, and the fact we are using two-photon excitation allow us to reduce the background so to have a better contrast. Bachmann demonstrated, but there are not many papers on this, so please have a look on this paper, and if you can find other papers on this, use the intrinsic fluorescence of DNA to get super resolution. Fine. Since with two-photon excitation we have a very high level of illumination, we can also take advantage of what is called second harmonic generation. So in this case, you shine light, you can get fluorescence emission, but since the light you shine has a very high intensity level, you can also promote in some samples and for some architectural organization of your biological sample an emission at 1/2 of the wavelength you're using. So if you're using red, you can get blue. That is label-free. We demonstrated this in the backward for the first time in 2002. And here you can see what's going on with the tail, for example, of a zebrafish. The green information is the fluorescence. But if you want to be sure, anatomically speaking, from which region this comes, the purple information that is the second harmonic generated information tells you anatomically from which part of this tail comes. There is collagen, and there are other things that can produce second harmonic generation. And so in this case, you can use autofluorescence forward and backward second harmonic generation to have a label-free view of your sample. Again, this is very interesting, but we are using a very high quantity of light, or we have to deal with a lot of background noise in case of intrinsic fluorescence. For this reason, again, my granddaughter is asking, are there other ways for performing label-free? Come on, how can you say no to this guy? So the answer is yes. We go to the coffee machine, and we think that when we are at the coffee machine, we do something that we can play, that we can do with light. We discussed this many years, I discussed this many years ago in 2 papers. When you go to the coffee machine, you orient your coin in the coffee, so you are polarized. In case you try to insert your coin 90 degrees with the aperture, you don't get any coffee. In case you throw your oscillating coin, you can get a coffee by chance. So why not using also adding another information, so polarization, inserting the light or the coin in the right way? Well, this is comparatively easy, so we are able to manipulate light and to control polarization states. It's an electromagnetic field, so we can play this game using devices and using our control of light. But what Mueller brought to our attention is a very nice way for playing this game in an ordered way. We have a vector telling you about state of polarization of light. You see is called the Stokes vector on the right side, tells you about different states. You use describing light using this vector also in the output. But if something happens between the input and the output, responsible is the sample. And since these are two 4 elements vector, you need a 16 elements matrix to describe what's going on. If you have known optical elements, you can put these in a kind of linear chain that you see colored down. But if you have an unknown sample, you can try to describe by each of these elements what's going on. You can have refringence, dichroism, depolarization. So you can have 16 images at the very same time, simply detecting and illuminating the sample with polarized light and detecting specific state of polarization. In this case, it's birefringence. In case of chirality on the surface of this beetle, is a chiral object. We demonstrated we can use this to get interesting information, label-free, during the development of biological systems. Dichroism. The very first idea came watching circular dichroism, that is differential absorption of light. But we want to work outside the absorption bands. And Carlos Bustamante had his thesis work in order to understand if we could get information from chiral objects. And starting from this point, so from the analysis of circular dichroism outside the absorption bands, so scattered light, we started using this approach for detecting different order of organization of helices in superhelices. Now you know better than me that chromatin is organizing in superhelices the helix of DNA. And so we decided to build an instrument working on this principle, and we decided to use the very same light source for performing two kinds of contrast, for getting two kinds of contrast from our image. We use a titanium sapphire laser to excite under 2-photon fluorescence, and the very same line is used to, and here we shape the polarization, to detect Mueller matrix elements. I have to say, just only telling you, that in order to control polarization, we have different methods, and now we are using something that we decided to buy on eBay for 300 euros. That is a Zeeman laser. So the output, you have controlled circularly right and left states. We performed some simulation in terms of light matter interaction with superhelices in order to be sure that we could detect differences in terms of organization of DNA within nuclei, so cells, and also on isolated samples. We also played this game with viruses in droplets of water. Sorry for going fast, but I want to go to the intelligent part. And so if, in case you are interested, we can discuss about this. But in the cell cycle, this is relevant from cells to molecules. At the interface, you know that you have chromatin organization, a different level of compaction in the nucleus. So different level of compaction means different superhelices. Are we able to detect them using a label-free approach? Yes. And we compared this with fluorescence just to be trained to understand what we are watching. And so now in our architecture, MOMICS architecture, we can definitely add the label-free part that we can combine with the fluorescence one. And this is MOMICS. Chromics in case of chromatin. And I bring to your attention chromatin, not only because I'm interested in chromatin, because you know probably better than me, but the situation is rather complicated. You have so many data, so you have many chances to get information. You have so many data. And since you have so many data, you can add the element that also Marta had in mind, Marta Fioravanti, that is artificial intelligence. I mean, having a lot of data, using them for training a machine is interesting. But what we are interested in is two steps in the intelligence. The very first one is reducing the number of parameters we need for taking a decision. The other one is to use these parameters for deciphering what's going on. Nautilus has a very rudimental nervous system. So in case a predator comes, if you provide a scene like this one that is rather complicated, is not able to take the decision that a predator is coming. For this reason, nature decided to provide Nautilus with a pinhole that is simplifying the information to the nervous central system, and so Nautilus is able to play the game. This is what we want to do with sound calculation, and Lisa Kuhn in our lab suggested to do this using independent component analysis. This means that we want to see things in a different way. And we decided to use independent component analysis, tensor independent component analysis, so TICA, to reduce complexity. So we provide the system with multimodal information. The system extracts parameters, so reduces the complexity. And then we can train the system using these parameters for understanding if you are in a healthy case or in a pathological case. And also we want to improve the data set in terms of multimodal data set using our ability of detecting label-free based information. So what we do is a two-level job in terms of artificial intelligence. One is to build a system able to extract parameters. Then we use a convolutional neural network to take advantage of the information we get. Convolutional neural networks have been used, for example, to get super-resolved image after some training. Now, I don't go into detail, but this set of multimodal information can be used at two levels. You see here, on the left side, you extract parameter. In the center, you have the parameters, and now you have a supervised actions that are, you provide now samples, for training the system using those parameters. And in case you are able to detect among the different cases, it's done. Otherwise, you have to go back to the extraction of parameters for getting the new training. Now, for the training, you need a lot of data, and probably you know better than me. But when you get this large number of data, you have a problem with this kind of approach. You lose the control of information in the meanwhile. So you cannot go back understanding why some parameters have been selected or not. Forget about it. Think only about the final result. The final result we want to get is we train the system with a couple of label-free and fluorescent image. At the end of our training, the system will be able to transform a label-free image in a fluorescence image, and we use this image for training, and we use a known system, like in this case, these tumor, malignancy progression, and chromatin remodeling, to train the system in order to be able to understand, to distinguish between healthy and pathological cases. At the Allen Institute, they played in a very nice way this game, and so they collected, they're using a database having a lot of label-free images, in this case collected using phase contrast. So we can discuss about this in case you're interested. And from this data set or transmission, they are able to produce a data set of fluorescent images without labeling. Like in the case you can see here. It's a long way training, but you can play this game. On the Nikon, you have a system called Convert AI that plays a similar game. So our system, our machine, in the inspiration of this graph, is made by the conventional microscope with all the possibilities we have, plus an artificial intelligence network that is the new component of this system that allows us also to manage label-free fluorescent image and to make our machine universal, plus an additional step that is called liquid tunable microscopy. I want to show you this related to the MOMICS exhibition. And so the name is the same of our microscope, because here you see a complicated real world that is depicted by few blue images. So we want to use the blue images to go down to this dynamic world using the microscope. So we want to go behind the visible, beyond the visible. In case you are interested, there are these two books reporting to this, and maybe you know the Petit Prince book that tells you that sometimes you see something you recognize here like a hat. But if you have a better look, you can recognize it's not a hat, it's a snake eating an elephant. Now, my granddaughter is asking about the future of microscopy also, maybe you, I'm sorry, I'm late of 2 or 3 minutes. My point is multimodal optical microscopy boosted by AI, fluorescence dynamics, so using the information coming from the different light time, changes of wavelength, changes of intensity due to the presence of molecules around the fluorescent molecule we're interested in, label-free using the Mueller matrix or phase contrast or transmission, plus liquid tunable microscopy. Liquid tunable microscopy because you have an instrument, classic, in this case writing machine, but you have to add some new view, like in the Aleph. Really, you can see whatever you want using the microscope. And you can combine together your information in a different way from the expectation of an image that you have in your mind. Sorry for the Italian, but are you sure that this is not a floor or a ceiling? This comes from Escher. Are you sure that when you see this image, is not another image, like in this case in the Galilei Museum, bringing to you the information? So Liquidopy wants to provide the opportunity to see things in a different way using artificial intelligence. An image where you don't recognize a cell, but you can find the parameters for understanding if you are in the healthy case or in the pathological case, or you can predict if an healthy cell will become a pathological one. All these things are possible due to this group. That is a great group, and also I am indebted with Partha for the time spent with us and developing with us some approaches. I thank really for your attention. Sorry for being 4 minutes out from my schedule at the time. And so I hope that in the lesson of Gregory Weber, you can turn a new page. And this is for those who put the doubt above the belief. Thank you very much for your attention, and I'm available for questions.
Yes, thanks a lot, LB, for this nice talk. And this talk is such a blend of, I would say this is the artistic talk that blends art and science. And I haven't heard such a talk or pretty talk before. And I think you have done it for the first time. I don't know, but I'm hearing this for the first time. I've heard your other talks. So SPAD, array modules that you talked about is really a good one. And we want to know a little bit more. And many labs, I'm sure, around the world have benefited, are going to benefit from this talk, which is being watched in most of the labs, as I see in my lab as well. And I'm happy that I'm able to host this talk, particularly this talk, and for a national program like Lab Visits, which is going to touch a lot of students around the country as well as internationally, I believe, because this is all through YouTube Live, and it's all through. And this talk is going to be watched again and again. I don't know how many hits you will get. In future, you will get a lot of hits. Okay, so I guess it's time that we ask a couple of questions if some of them want to ask. Who are
and sorry, Partha. Let me also say that all your students and you are more than welcome if you want to come visiting our lab or collaborating with us for improving the Momix microscope.
Thanks a lot, LB. I mean, we are also looking forward to that. So, if people, anyone who want to ask anything. Okay, so I guess LB, I have something to ask about this. So you talked about SPAD array detectors. I was wondering what is the quantum efficiency and the temporal resolution of these new detectors. If you can let us know.
Well, in terms of temporal resolution, I think that now, for example, with the image scanning microscope using the SPAD array, we are not limited by scan rate. So you can really go very fast. I didn't mention in this talk, and I'm sorry that I brought to you so many things all together. I didn't mention that today we are also using a liquid lens. That is a lens allowing us to collect information at a very high rate. So we can collect today, I think, 2000 frames per second, also in the 3D. And you could say, okay, but what about the photon and what about, noise? Because if you are fast, you collect a poor number of photons, yes, but we can be fast passing through the very same point several times, and so we can increase the budget of information. Today, our temporal resolution in terms of spectroscopy, so in case of using the SPAD array in the image scanning microscope, is down to 100 picosecond. So we can avoid using a colored filter to understand which kind of fluorophore we have. And in terms of speed of imaging, we can go normally down to 100, 200 frames per second on a 512 image. So you can follow dynamic events in the cell in 3D. And you don't waste photons because you collect a good number of photons that you can rearrange and you can reassign in case you are using image scanning microscopy. But also when you are using STAD today, since you are also using the lifetime information that comes for free. I mean, so you have not to say, I want to collect it or I don't want to collect it. You have only to say, I want to use it, I don't want to use it. But you can collect this at the conventional frame rate. So you can move from in terms of temporal resolution, from a very fast resonance scanning microscope up to a large number of frames per second. So temporal resolution is not a limitation now.
Yeah. Okay. Thanks a lot, Alvi, for all the details.
Thank you very much for your
Yeah,
passion.
we are not getting any more questions. So, once again, I thank you, and thank you for helping us.
Okay, and I will send you
program.
I will send you the PowerPoint so you can distribute in case you are interested. Okay.
Yes, that would be great. That would be great.
Okay.
Okay. So, yeah, thanks a lot, Alvi.
Thank you very much. Ciao, Parta.
Bye.
Hi, everybody.